Olga, the floor is yours.
Thank you. I hope you hear me fine. Can you give me like
Yes.
some kind of indication?
We can hear you. Thank you.
Okay, great. Thank you. So thank you for inviting me to give this talk. So I know originally it was planned to be a little bit more high resolution than I can provide, but I think it'll be also relevant for you. So I will today I will briefly introduce you to X-ray microscopy and imaging at APS, which is relevant to biological and environmental sciences, which beamlines conduct this research, and we'll share a few science highlights with data which use data generated at our beamlines. So Jocelyn had very nice animation of how X-ray fluorescence work. I do have, oh yeah, by the way, let me try and get the pointer. There you go. So yeah, so I have a cartoon. So basically the way X-ray fluorescence works, if you have some X-rays and you apply it to atom, as long as the energy is higher than electron binding energy, that electron may be removed, and then upper shell electrons will go down, and this process will result in release of another X-rays, which will have energy specific to that atom. That energy can be measured by the detector, and when detector is calibrated, we can say based on the value of that energy, what kind of atom emitted it, and based on the amount of photons measured, we can correlate to amount of those type of atoms in the sample. So moving on to scanning X-ray fluorescence microscope, when we have synchrotron, we can generate a lot of X-rays. So when we have this huge amount of X-rays, we can use optics like zone plate or KB mirrors, which will squeeze X-rays into tiny spot below 1 micron, or maybe like right a little bit above 1 micron, but the very tiny spot. This process of focusing results in a loss of photons, like sometimes relatively dramatic loss of photons. So that's why super high resolution, super tight focusing makes sense with synchrotron X-ray sources. Wouldn't quite work with tabletop devices. And then we can basically measure the same kind of photons from emitted photons, putting detector perpendicular to the incident beam from the tiny spot in the sample. And then we can move the sample and measure another spot and another spot and another spot. And then we can build the image, elemental image of that sample. And if we also have detector on the path of the original X-ray beam, then we can measure diffraction or scattering or absorption of the sample, and then we can have multimodal imaging in this case. So this technique is quite highly sensitive to detect like a whole bunch of elements, but it has some limitations. So the main limitation for biological sciences is we're kind of blind to such an important elements like carbon, oxygen, sodium, and so on. So we start very nicely, precisely detecting somewhere about silicon or aluminum, depends on the setup. And then we can detect everything, all the essential elements and trace elements above aluminum and silicon. And we can also detect higher Z elements, which will produce higher energy emission, such as platinum or gold, lead. Some of those elements, they should not be in biological or environmental science systems, but sometimes they end up in those systems either due to pathological exposure or intentional placing of those elements to visualize something like gold nanoparticles or something like that. In addition to that, synchrotrons also offer such imaging techniques like tachography, which can be producing images in 2D and 3D. It is basically produced using those overlapping scans over the sample, and the result is a high resolution imaging of structural variations of the sample. So here is an example of Chlamydia mona algae, where on the right, we have tachography, and on the left, we have elemental distribution of a few elements in there. And in addition to that, we also offer full field imaging, which can be handy for a variety of samples. We have 3 beam lines which offer this technique. 2 of them offer micro CT, and 1 of them is capable of doing nano CT imaging. And ideally, we want to creatively combine those techniques to get the maximum out of the sample we work with. So what do we work with? So our samples vary from size to size. We work from like single cells and even organelles within the cells. And we, here will be increasing the field of view and resolution. So we can go to organoids, which are like miniature 3-dimensional organs. We can do tissue sections, and sometimes we work with fossils, and we cannot fit the full T-Rex in the beam, but sometimes we can fit just one arm for some of the very interesting paleontological studies. So where do we get our samples? We go up in the air and study aerosol particles. We look at the soil aggregates. We look at the plankton and fish embryos to study distribution of like natural distribution of the elements within them, as well as redistribution of those in case of certain pathologies. And we look at the microbial cells on the top, like on the surface of the water, and deep down to the bottom of the flumes. So we also work with animal samples, again, starting from cells, organoids, tissues, and full organs sometimes, depending on the needs of our users. And in terms of plants, we look at the roots, we look at the stems and branches, and we analyze flower buds to figure out distribution of certain elements and how they affect plant fertility. So samples can be mounted in variety of sample mounts depending on which beam line we use, what are like physical constraints of the beam line, and what is reasonable for the system we work with. So we usually use silicon nitride windows, like you see here, for single cell imaging. Also TM grids can be used for that. But they may provide stronger background, and those are mounted on this kind of aluminum sticks, which can be inserted into multiple beam lines in case if you need to have a different kind of resolution from your system. We also have like slightly bigger mounts for the samples, which are mounted on Kapton film or like ultralin film. We can accommodate crystallography support samples, like larger slide holders, riser boxes, and different kind of sample cells. So the beam lines, which were capable of this imaging, which is useful for, usually used for biological and environmental samples. So I am a member of microscopy group, and we have several beam lines at sector 2, right here, at 8 BM, and they will stay there after upgrade. We used to have beamline at sector 9, which moved to 2 as well. We also have nanoprobe, which is usually used for material science samples, but some biological samples can be imaged there as well. And other beamlines of APS, which are part of different groups, include 13 IDE, 20 ID, which will disappear after upgrade, and micro CT and nano CT I mentioned previously. And by the way, so these beam lines and micro nano CT beam lines are part of IBRLITE initiative, where we facilitate the access to the beamline for BER users. So after APS will undergo upgrade, we will get better beam, which will give us way more flux than we have right now, and it enable new science for us, or the same science, but in much higher throughput or with much higher resolution than we can do right now. So for 8BM, for example, we will not only increase the resolution from 20 microns to 5 microns, we will also use a different monochromator, which will enable faster scans, and we will have X-ray fluorescence tomography. And here's an example of one of the science cases we had from this beamline, where we were studying mineral depositions in the layers of the beluga whale tooth. 13ID microprobe, they have variety of techniques available. And here I'm listing the beamlines in, starting from lower resolution and going to higher resolution. So they have variety of techniques available, including fluorescence, spectroscopy, microdiffraction, and tomography. And they will upgrade their optics. So they will go to submicron resolution and will be able to do cryogenic sample preservation. And here's an example of one of the projects from the recent projects from that beamline, which was used for analyzing the pores and roots within the soil sample. 2ID beamlines, we have two branches. They are kind of close, but not like fully identical. So one of them has a finer beam and slightly larger energy range, which kind of determines which elements you can reach using this one. It also has microxanes and tachography with cryogenic sample environment. 2IDE will have a cryogenic environment as well. Previously, it had slightly larger beam, larger flux due to different monochromator, and we could do tomography and in situ experiments. And after upgrade, we aim for higher throughput. So we're not gonna push resolution too far. So we're gonna improve it from 200 nanometer to like maybe 50 nanometer resolution, but we will have much higher flux and will go much faster. What it means that if I work with a soil aggregate to generate this kind of image, which is basically like few slices from the full aggregate, it took me about 36 hours for this kind of slice. And after upgrade, in the same amount of time, I will be able to image the full aggregate. By the way, yeah, so that aggregate was colonized with bacteria, and that's why we see different colors in there. So part of it is aggregate, and part of it is traces of bacteria going through that aggregate. So BioNanoprobe has very high resolution beam, down to 30 nanometers. It has cryogenic capabilities, which is very important for biological and environmental science samples. And it also has a tachography as one of the components. So that chlamydia image this chlamydia image was coming from that beamline. And after upgrade, they aim for much higher resolution of 10 nanometers. And they will also improve their tomography stages to get better quality tomography reconstruction and add a capillary to improve their signal. So now I'm gonna describe a few science cases out of many we have. So I'm gonna start with like the larger sample and gonna go to the higher resolution imaging. So one of the samples I wanna describe is a project I work with Ken Kemner for like many years right now. We work with imaging soil aggregates. Soil is an important medium for supporting plant growth, important for nutrient cycling and so on. And it's very, very interesting subject to study and kind of difficult due to its opacity. So to analyze the way how microbes can migrate through the soil, those microbes were fed quantum dots. and they were glowing in green when they were checked under microscope. And just to keep you a little bit more confused, when we imaged them, we colored them blue for some reason. So those quantum dots had selenium, and we were able to image that selenium when we were scanning soil aggregates. So on the left, you see the image from micro-CT. And on the right is just a two-dimensional image from X-ray fluorescence beamline, where blue is selenium and green is calcium, iron is red, and you don't quite see them on that left side due to self-absorption artifact. And as a result, there is this reconstruction of the slice, and what you can see here, it's not the real-time like bacteria colonizing that aggregate. It's just the gradual increase of intensity from selenium, showing how those bacteria travel through that slice. So yeah, going higher with resolution, we study uptake of certain nutrients by plants, not only nutrients, also toxic elements. In this case, the uptake of phosphorus was promoted by endophytes. It was a project with Tamas. So here we were able to detect the difference in the phosphorus uptake for control and exposed samples. Other project, yeah, in this case, the samples were fixed and embedded for analysis. For my other collaboration with scientists from Canadian Light Source and University of Saskatoon, we were working with plants which were exposed to arsenic. And those plants, so those were controls, and some of them were PN2 mutants, which displayed low arsenic accumulation and transport. Those plants were grown in the sterile medium with controlled exposure to arsenic after a week of growth. And those roots, like if we're lucky enough and fast enough, can be extracted, sealed, and imaged while they're still hydrated, when we can actually see the channels inside the root. In addition to that, we also have quite a lot of work with wood, which can be like divided into big parts. One is visualizing distribution of additives to the wood. Like in this case, the wood was treated with epoxy. Epoxy had some chlorine, and we could actually do three-dimensional visualization of impregnation of wood cell with a chlorine, and therefore with epoxy. We also use this method to visualize the quality of impregnation of different medium for wood preservation strategies. Yeah, kind of refers a little bit to Jocelyn's talk here. We were also visualizing fungal decay in the pine wood. In this case, wood samples were incubated for 2 weeks. They were exposed to brown rot, and then changes in the distribution and just in general content of certain elements were visualized using X-rays, using X-ray fluorescence microscopy. In this case, we actually were using 3 different beam lines to look at 3 different resolutions. 8BM results are not shown here, but they were looking at like larger chips. Here, we were looking at 2-micron thick sections with fungal hyphae displayed here, and observed like dramatic changes in zinc, calcium, iron, and manganese. And here is the results from the bio-nanoprobe beamline, where they were looking at the hyphal anastomosis, like aggregates like this and like this, from what we see at 2ID beamline results, to see what it's made of and what kind of elements you can usually find in those. Yeah, unfortunately, at that point, the chemical speciation was not available, so we cannot quite see what kind of potassium we have in those samples. In addition to that, we look at single cell imaging or tissue imaging. So in this case, here is an example of Toxoplasma gondii study where we were looking at the importance of vacuole ion transport for parasite intracellular iron content and how that affects iron sensitivity for this parasite and as well as the survival and virulence of that parasite. So we were looking at the free parasites as well as the parasites which were infecting mammalian cells. And last but not least, we also look a lot at algae and plankton. It's ongoing collaboration with Benjamin Twining from Bigelow Laboratories. So plankton is very, very important for carbon sequestration. It depends on iron availability. It kind of feels like endless study, but it generates a lot of data which can be useful for different kind of modeling of carbon sequestration, oxygen production by the ocean. So here's just one of the examples and how we can visualize different fragments of the cell. There are other bulk analysis methods available to study this kind of cells. However, they cannot differentiate between organisms, or like if you need to differentiate organisms before you study them in bulk, you may introduce significant artifacts and so on. So it's proven to be like a very reliable tool to understand differences in nutrient uptake by the plankton in different zones of the ocean at the different times of the year with a different kind of pollutant presence in the medium. And recently they published a paper in Nature where they kind of accumulated several years of studies on this subject using data provided by synchrotron. And if we have enough time, we can also do tomography on this kind of marine samples. We can visualize and segment components of the microbes, cells from the ocean, or we can even visualize in 3D. Zebrafish embryo, like this one is just the native zebrafish embryo. We can see how these distributions of zinc, iron, and copper will change in case of, let's say, mercury exposure or like excess of iron or like any kind of exposure. However, in this case, 3D visualization takes a lot of time. So on average, if you would need like about an hour or 2 hours or 3 hours, depending on the size of the sample and the resolution you're looking for. If we want to do tomography, we need to do at least 60 projections to visualize what we want to see, to build the three-dimensional map of your sample. So it may take hours and hours, and compromises result in poor data quality. However, when we will finish upgrade and all the beamlines will get much more flux than we have right now, this kind of experiments from proof of concept will turn into routine operations, enabling us to answer much more interesting science questions than we have right now. So as a summary, I would like to list the beamlines which are available, which techniques can be used for biological environmental science studies. So most of them have X-ray fluorescence available. they're listed from like low resolution to high resolution possible. Some of them are multimodal, so we can do X-ray fluorescence and diffraction or tachography. And also there are 2BM and 32 beam lines which offer full field imaging option, which can be very handy like to begin with when we try to tackle complicated problems for which we need to do tomography, because those are much less time consuming than X-ray fluorescence or tachography measurements. After upgrade, we expect high performance metrology and scanning and control systems. We will improve our workflow for multi-beam line imaging and improve sample transition from one beam line to another. We hope to use higher count rate detectors, and we are also updating our reconstruction and data analysis software. So we're not going to have bottlenecks in these areas. And with this, I would like to thank my group I work with, like all the scientists which contributed to the instruments, results from which were displayed here. Also other APS members, Matt, Zu, and Victor. Zufin Frog is our contact person for Eberlite activities. Arthur, who made a huge difference in X-ray fluorescence data processing, and our engineers, which keep helping us build our instruments better and faster. And with that, I would like, and our finding source as well. With that, I would like to take questions if you have any.
Thank you. We have time for several questions. Are there any, audience? There's an online question?
There's an online question.
Do you consider radiation damage in data collection?
Yes, so in most cases, our samples come fixed, so we wouldn't see much radiation damage, but obviously when we go with a high resolution image, when we increase flux density on a certain pixel, we observe radiation damage, especially if you keep running, if you want to see, you run the same sample multiple times until you start seeing effect of radiation damage. So cryogenic capabilities help to solve those problems for majority of biological samples. It's not always optimum sample treatment, and then in this case, you kind of take what you can with a pinch of salt. But cryogenic will be the best option to preserve the sample to prevent radiation damage.
Thank you. Questions? If I may ask, I'd like to follow up on the what happens after the upgrade. You mentioned that data collection becomes faster and also data processing. So, for example, when you scan, you mentioned an aggregate that was scanned by XFM tomography, and you mentioned that instead of some fraction of it, you can scan the whole thing in 10, 24 hours. So by how much time, how many times it would be faster, like 5 times, 10 times?
So it depends on from beam line to beam line. So for some, we expect 5 times higher speed, for some of them, 20 times faster, and so on. So at some point it will become, right now, X-ray fluorescence experiments for biological environmental science samples are flux hungry experiments. So we don't have enough flux. We spend for let's say, plankton, we spend sometimes 5 seconds per pixel, which is a lot. So after upgrade, we will go faster. But then in some cases, for some of the samples, we will be limited with a readout speed of our detection system. So in this case, that will be our bottleneck, not the amount of photons we put on the sample. So yeah, but depending on the system, we expect from 5 to 10 to 20 times faster.
I heard that basically on the fly reconstruction of micro CT data is available, like using high performance computing. Will that be available for XFM tomography as well?
We work on that. At this point, when we don't get big surprises from the spectrum and everything is pretty much straightforward, then yes. Still, every once in a while, the straightforward spectrum fitting doesn't give the results we like. So in this case, it will take longer. But for like typical for typical biological samples, I think we will go with automatic data processing. Yes.
Any new questions? And let's thank Olga again.